Okay, so good afternoon, everyone. We're going to dive right into topic 2 here in a moment. So topic 2 is on macromolecules. So topic 2, if you take a look at the textbook references, is kind of focusing on chapter 5, but little bits and pieces from chapters 2, 3, and 4. So if your chemistry is a little bit rusty, then you may want to brush through those chapters a little bit. If you're kind of fresh with your chemistry, then you probably don't need too much of a review. Before we get into this topic, I just wanted to make an announcement about the lab for a moment. I've been working hard on getting the virtual lab stuff prepared. So I'm like 90% done. So the lab instructions will be sent out probably tomorrow. I'm hoping by noon, it could be 1 o'clock. And you'll have an idea of what you'll be doing. The other thing to know is I will also have a Zoom link that goes with the lab, and I will be available from 2 o'clock to 4 o'clock tomorrow, or yes, tomorrow, for anybody who wants to join into the meeting at any time. Use it as kind of like an office hour, or to discuss the lab and the assignments. You might not find you feel the need this week, but maybe in future weeks once the assignments start coming in, and you want to either discuss your reports in particular, or if you want to discuss the lab exercises and those kind of things. So I'll have that information sent out tomorrow, and so please use the time tomorrow afternoon to work on the lab. This first week should be relatively light, and hope you enjoy what you're learning so far. Okay, so let's talk about those macromolecules. So firstly, a little bit of chemistry. What is a cell made of? You probably know mostly made out of water, about 70% water. Kind of depends on if you're looking at a plant cell or an animal cell or whatever, but about 70%. A small percentage of the cell is small molecules, ions, solutes, things like that. And a big percentage of the cell, the dry weight anyway, is made of macromolecules. So here's the rough breakdown. We got proteins, RNA, DNA, polysaccharides, and phospholipids. There are other lipids in there again. Most cells don't have a huge amount of lipids in their cells other than the phospholipids, unless they're fat cells. So here's a little portion of the periodic table of elements, and if you take a look, this is showing all the elements that are believed to be essential for human life, and there's a few that might be essential that we're not sure about yet. But if you take a look at these ones here, so hydrogen, carbon, nitrogen, oxygen, this is about 90-something% of all of the compounds in our body are composed of these few elements. So if you end up taking organic chemistry next semester or next year, you're going to be focusing on these elements, and you're going to learn a lot about their chemistries. So you probably know a little bit about their chemistries already. These are some, I guess you'd call them Lewis dot structures of these elements. We've got hydrogen, oxygen, nitrogen, and carbon, and it's showing their valencies, how many bonds they make. If all this is unfamiliar language to you, then I do encourage you to spend a little bit of time reviewing the chemistry in the first few chapters of the textbook. So here's where it's starting to get a bit more important for this class, is that you can start linking these atoms together and we can get various functional groups. So here are some functional groups that are going to be worth knowing a little bit about, at least for this class. And again, if you take organic chemistry or biochemistry, you're going to need to know these things inside out and backwards. First group is this hydroxyl group, or the alcohol group, which is an OH. You're going to see a lot of those in carbohydrates. Second is carbonyl, also found in carbohydrates. You can see it's a C double bonded O. We have the carboxyl group, or carboxylate group, which is found in carboxylic acids. So could be found in fatty acids and amino acids is where we're going to find this one. I'll point these out, by the way, when we're looking at the macromolecules. Here's the amino group found in amino acids, sometimes nucleotides. Sulfhydryl group found in one of the amino acids, and the phosphate group found in nucleotides, and also phospholipids. So these are the 4 groups of macromolecules we're going to be talking about: lipids, carbohydrates, proteins, and nucleic acids. Kind of like the 4 food groups. So let's, before we take a look at these, I want to talk about something, how these things are linked together and why we're calling them macromolecules. If anybody knows what the word macro means, you probably know macro means big. So these are big molecules, and most of these big molecules are polymers. So what is a polymer? Polymer, poly means many, mer means component. So polymers have a bunch of components attached together. They can be identical. So you could have starch, which is just a bunch of glucose residues, or you could have something a bit more complex, like amino acids. You've got 20 amino acids linked together in different sequences. So how do we link these polymers together? We link them from something called monomers. There's another word for you: polymer and monomer. And so if you take a look, here is a typical polymer. You can see it has an OH, that's an alcohol or hydroxyl group. And it's got an H on the other end, and here is the monomer. So if you're a biochemist like I am, you kind of look at that and I see an HHO, and that's H2O. So it turns out we can link these 2 things together by removing that H2O. So if you do that, that's called a dehydration reaction. Some textbooks call it a condensation reaction, but I'm going to stick with dehydration because that's what our textbook is using. So there we go, they're linked together, and now we have a longer polymer. So it turns out that this entire reaction can be done in reverse. So let's do that. Oops, wrong direction. Try that again. The reverse reaction is called hydrolysis. So if you take a look at that word hydrolysis, hydro means water, and lysis means breaking, so water breaking. So the reverse reaction, we're adding water to that polymer and making it into 2 short polymers. So you're going to see this reaction again and again and again, by the way, if you take a lot more biochemistry. Okay, so I want to just try to, as in the next few units, start introducing some kind of sample midterm questions. Might be a little bit different this year because it's all online, but traditionally I've done a lot of definition type questions in my exams. And why do I do definitions? Because we're going to learn a lot of new words and want to make sure that you do know what those words are. So my definition type questions look something like this. Usually what I'll do is I'll have, let's say, maybe 8 words and you pick 5, right? So they'd be worth 1 mark each. So here's an example: define polymer, and here is the definition I got from the textbook. And you can see what I'm looking for is not a 1-word answer. I'm not looking for a short answer. The answer does not have to be a complete sentence on my exams, but to get the full mark, it does need to be a complete thought. So in this case here, I've broken up the definition into 2 parts. You can see a large molecule. I probably might give you half a point for that, but really this other part here about consisting of many similar or identical building blocks, that's kind of important, and linked by covalent bonds. So you're probably wondering about polymers. Aren't plastics polymers? Yeah, a lot of plastics are made out of repeating units of, like polyethylene, for example, is made of repeating units of ethylene. Polypropylene, made out of repeating units of, I think, what is it, propyl ether or something like that. I have to look that one up. Okay, let's talk about lipids. Lipids come in many shapes and varieties. First one I want to point out are these ones called fatty acids. So if you take a look here at, I'm just going to grab a pen here, and there we go. Take a look at this right here. This here is a carboxylic acid group. The other one over here on the right has one as well. So those are carboxylic acid groups, and so these are acids. But the rest of the molecule is the fatty part. So, you know, it's just coming from our dietary words when we talk about fat, right? And so that's the lipid part. And if you take a look at the rest of the molecule, it's carbon, hydrogen, carbon, hydrogen, hydrogen, carbon, It's all carbon and hydrogen. And if you remember your high school chemistry, carbon and hydrogen have very similar electronegativities. So most of this molecule is non-polar. And that's a very important definition when we're looking at lipids. We have some other notes on this slide. There's the notes. You can see carboxyl group or carboxylic acid. That's polar, but it's just a small, teeny, tiny part of this. Most of the rest of the molecule is this long hydrocarbon, which is non-polar or hydrophobic. So there's that word again, hydro, right? That means water. And then phobic means fearing. So hydrophobic means water fearing or not mixable with water. I want to just point out the one on the right is a little bit different than the one on the left. So what do we have here? One on the left is called saturated and saturated with hydrogen. So that means it has this hydrocarbon portion is all single bonds, and the unsaturated has some double bonds. So those double bonds make the molecule kind of kinky in a way, meaning it's kind of bendy. It doesn't have a nice uniform kind of zigzag shape. So what's the consequence here of these fatty acids being unsaturated? Show you another picture here. The saturated ones, if you look on the left, so I'm looking right here, these stack really well. And because they stack really well, there's a lot more close London dispersion forces, right? So they can pack a little bit better, and they're often solid at room temperature. So when something's solid at room temperature and it's a food, we call it a fat. If you look at the one on the right, the molecules, like I said, I call them a little more kinky. They're bendy. They just don't lock in together as easily. So they slip around a bit more, and they're often liquid at room temperature. So we often call these oils instead of fats, but they're all lipids. So lipids, fatty acids, I mean, here, let's go back to this for a second, don't usually exist just as fatty acids in our diet or in our body. They usually exist in something called triacylglycerol. So you can see that word is up here, triacylglyceride. Or sometimes people call these TAGs. Triacylglycerides. So the glyceride or glycerol part comes from here, glycerol, this little molecule here. So glycerol has 3 carbons and 3 alcohol groups. So 1, 2, and 3. And the tri part is we're going to add 3 fatty acids to this. So here's a fatty acid. You can see the carboxylic acid there, and we've got another H2O that can come out. So we're going to use that dehydration or condensation synthesis, and this fatty acid is going to be added to the glycerol. So we'll add it, and then we'll add 2 more. So there we go. So now we have a triacylglyceride, tri meaning 3. So this is what we call a fat molecule, particularly when we talk about dietary things as well. We talk about eating fat, right? And this is usually how fat is stored in plants or animals, or if you eat too much, this is how it's going to be stored in your own body. So there is some fat in some adipose tissue. So adipose tissue is human fat tissue, is what it is. And you can see it's not really that soluble with water. It's forming these big blobby things. Okay, so I'm just going to take a quick break here from the PowerPoint, and usually what I do as I go along and give my lecture, I'm usually writing a lot of notes on the whiteboard, so I'm going to be, I guess instead of the whiteboard, using this Word document. I will not be sharing these Word documents with you. So if you want these notes, you'll have to take them as you're attending class or find them later. Some of them are going to be like this, which will be a summary of what we're talking about as we go along. Other times there'll be new notes that you're not going to find in the PowerPoints. And usually I'm trying to emphasize some things that are important. Okay, so this is not going to be complete after the lipids part. I'm going to come back to this several times and add to it. So you can make a similar table if you like. You can add your own notes, but this is just giving you an idea of what this is going to look like. So you can see I have lipids, and what are the basic units? So the basic units are triacyl. The basic units are fatty acids plus glycerol. And you can see there's a thing I have there under linkages. Let me just go back to the PowerPoint for a second. So if you go back to this PowerPoint, I just wanted to mention that a lot of fats have these ester linkages. I don't want you to get caught up in the chemistry of that, this ester linkage here, too much. When you take organic chemistry, you're going to learn more about ester linkages, but it's just a common type of linkage that you would find in fats. And you can see it's there, characterized by C double bond O and then another oxygen as well in there. Okay, so let me go back to the here. So I'm going to put that. So we've got ester linkages. By the way, it is not an ester linkage like that. That's a person's name. Just so you know, person's name has an H in it. If you're talking about organic chemistry, there's no H. So what are the functions of fatty acids plus glycerol? By the way, I'll just put tags there, meaning that it makes triacylglycerides. So you probably know that fat is food. It's a food source for us, and it's also energy storage. So we're not done with lipids. We're going to come back to this. Oh, maybe one other thing that I can add on to that is protection of your organs. Excuse me. What you may not know is your heart and certain other vital tissues actually have fat tissue around them to protect them. Excuse me. All right, so back to the PowerPoint. We're not done with lipids. So here's another type of lipid that we mentioned earlier on in the lecture, are the phospholipids. So the phospholipids, I want you to take a careful look at the structure of the phospholipid, right? We have gray over here, this gray thing. This is glycerol all over again. You can see the label glycerol. I have a fatty acid here, and I have another fatty acid right here. So two fatty acids, and the third fatty acid is replaced by something else. You can see it's replaced by a phosphate. Oops. There's a phosphate and also another polar group, in this case something called choline. So phospholipids are very similar to triacylglycerides, but they have that polar, extra charged groups on them. And so these have kind of some unique properties. Notice, I mean, I could draw this out every time. I just want to point out that the textbook often will, sorry, again, I can't find, okay. The textbook will often draw them kind of like this. You've got a little polar head and these two little tails that are hydrophobic, and you'll see that that symbol representing a phospholipid quite frequently. So these phospholipids, if you throw them in water, this is what you get. You can actually buy these things from chemical supply companies. You could buy, a kilogram of phospholipids if you wanted to. Throw them in water, and they don't dissolve in water. And they clump up, and you can actually see they clump up sometimes in these little round things, and sometimes they clump up in other sort of long, weird structures. And these have names. The round structures are called micelles, and the structure we're really interested in this class are these bilayers. So you can see what's happening with these phospholipid bilayers is that the polar portion, the phosphates, those are polar and they're hydrophilic, so they can interact with water. And the hydrocarbon tails are hydrophobic and they can't interact with water. So you have the phosphate heads contacting the water, and you have the hydrophobic tails making their own zone, and it becomes a phospholipid bilayer, which of course, if you probably know, is that this is a very important part of our membranes. So let's go back to our Word document, our whiteboard, and I will put that down here, a basic unit. We also have phospholipids. Okay, and so these are part of membrane structure. There we go. Okay, still not done with lipids. There's a couple other things I still need to say about lipids. Obviously, the ones I talked about are some of the really important ones. You can see here's our cell. If our cell is mostly made of water, what are lipids doing? Well, they're actually giving us a membrane to give something to separate our cell from its surroundings. Some cells have internal compartmentalization, so organelles, so they're also helping with that as well. And sometimes they're food or a food source or a source of energy storage. So there are other lipids out there. Here's another type of lipid. These are called steroids. So I found this picture on the internet. I still have not figured out whether this guy is real or not. But this is probably what you associate with steroids, is people on steroids and trying to build muscle mass. But steroid doesn't necessarily mean a hormone that's building muscle mass, although testosterone is probably the most famous one. Steroid or sterile compounds mean it has this 4-ring structure. So you can see 1, 2, 3, and 4 rings. So testosterone, of course, is a sex hormone found both in males and females. Males ending up with a bit more of it usually, so they can build muscle mass, and also does a few other things like adding body hair and things like that. There are lots of steroid compounds out there, though. You can see cortisol. Cortisol can be used as a drug to reduce your immune responses. So sometimes if you have eczema, or if you're getting some sort of infection that's causing you to bring fluid in your lungs, you might get some sort of cortisone. Sometimes people have inflammation in their joints and they're getting injections. Progesterone and estrogen are female sex hormones. There's a whole bunch of these steroid compounds out there. Cholesterol is another one. You can see cholesterol I have here in bold, and it's involved in membrane fluidity. So we're going to talk about cholesterol just a little bit in topic 4, in sex steroids and pheromones and all sorts of other things out there. Okay, so I better add that to our notes here. Okay, so we have steroids, and add that one. And what are steroids? Steroids are, we have hormones, membrane structure, and I'll just say etc. Okay, so back to my PowerPoint here. Okay, okay, so it looks like I have a Kahoot for you guys here. So I'm gonna get you to load up your Kahoot app, and I will load up the quiz. So where is it? Macromolecules, this one here. Can I give you about 15 more seconds to join? All right, here we go. Identify this molecule. Okay, so the correct answer is it's a fatty acid. So I just want to make a point and say that, it's important you be able to identify the different types of macromolecules. And I'll have a bunch of more examples to show you. I'll definitely have at least one or two questions on the midterm that will show you a few, and it'll say which one is correct or which one is incorrect or something like that. It is important that you do know some of the chemical properties over these things. You don't need to draw them for me, but you will need to be able to recognize them. So there's the picture. Why is this a fatty acid and not a triacylglycerol? Tri, remember, is 3, and we only have one fatty acid here in this case. All right, let's see how the points came out. 873 points. Good job. Question 2, another molecule to identify. Okay, good job. So this one was a triacylglyceride. One person guessed phospholipid. I don't see a phosphate on there. So there's no, can't be a phospholipid. So good job if you guessed triacylglyceride. Okay, so I have one more question for now. Okay, so this one here was indeed a phospholipid. You can see we've got the 2 long carbon chains and you've got that phosphate group there. All right, let's see the points. Okay, Shane is in the lead. Good job. So I'm just going to check the next question. I may stop it actually and go back because I may be saving the next question for a little bit later in the lecture. So hold on a second. True or false? Yeah, so this one here is for later. So we'll just skip and finish for now. Okay, so I'm going to go back to the PowerPoint, and we will come back to Kahoot some of these questions next day as well. So where's my PowerPoint? There it is. Okay, let's talk about carbohydrates. Okay, so actually, before we go on and talk about carbohydrates, I just wanted to go back and make a note about lipids. Maybe I will make the note up here. Okay, so this is kind of like a definition of what a lipid is. Okay, a lipid is a hydrophobic biological molecule. So that's my definition of a lipid, and I'm really sorry it's not more specific than that, but that is actually really what the definition is. And if you take a look at steroids and if you take a look at those fatty acids, the structures are very, very different. Some lipids are vitamins, like vitamin A. Again, there's a lipid and it's a very different structure. It has to be a biological molecule, so like something like gasoline is not a biological molecule, so not usually considered a lipid. Maybe I could expand that definition and say a hydrophobic biological hydrocarbon might be a better definition, because we're not talking about something like iodine, which is also hydrophobic. But anyway, I just wanted to put down that definition to say, hey, if you it doesn't just include fatty acids and doesn't just include glycerol and steroids. There are many other types of lipids. Carbohydrates, on the other hand, are a little bit more uniform in terms of their chemistry. There are specific things we can say about them. Not all of them are sweet like sugar, but certainly some of the yummy ones are. Some other names for carbohydrates, of course, are sugars or saccharides as well. So let's take a look. Here are some simple saccharides. So we call these monosaccharides, mono meaning one. And you can see we have, what do we have? Aldose, ketose, got ribose, ribulose, glucose, galactose, fructose. So a whole bunch of different carbohydrates. Take a look at them. They have tons and tons of alcohol groups on them. Pretty much almost every carbon usually has an alcohol group, maybe less one. So you can see they also have that double bonded carbon on there, which are both characteristics of carbohydrates. So these are not usually hydrophobic. These are actually, because of all those oxygens, very, very water soluble. You may be more familiar with the cyclized ring forms of carbohydrates. So you can see this is glucose here on the left, and it actually exists in equilibrium, in solution, in a cyclic version form and the linear form. So often you see carbohydrates represented as something like this. You can see this nice abbreviated ring structure form here. So we can link carbohydrates together. So if you take a look, there's glucose and fructose, and there it is again. We've got our H2O being formed, and we've got a dehydration reaction again. So if you take a look, you put those two things together and we have a glycosidic linkage. So I'll come back to that in our notes in a minute. So by the way, anytime you see this term here, glyco, okay, glyco means carbohydrate. I'm going to write that down. I don't have my pen today. I'm just using my mouse. So sorry, it's a little bit slow. Carbo. High. It's also not easy to write neatly with a mouse. This is not easy. Carbohydrate. There we go. So anytime you see glyco, remember that's carbohydrate. So glycosidic linkage means a carbohydrate linkage. So we can take these carbohydrates and we can link them up in even longer chains. So I'll go back to this slide, right? We've taken two monosaccharides and now I've made a disaccharide. We can make a polysaccharide if we add tons and tons of these things together. So we can make starch. So this is starch. So what we've done with starch is we've taken hundreds and thousands of glucose residues and we've linked them together. They sort of form a little bit of a spirally shape. So if you take a look here, then right here, it's a spirally shape, kind of like right there, like that. And sometimes we distinguish and we call starch amylose and amylopectin. Amylopectin, you can see, has some branches on it, but other than that, it's just starch. This is found in plants. So anytime we eat plants for food, often they're full of starch. So potatoes and rice and carrots are like 99% starch. And then if you are an animal, instead, we can store carbohydrates in the form of glycogen. So here's glycogen down here written. And if you take a look, glycogen is very similar to starch. It's just more branched. Makes it easier for us as animals to get at the energy a lot more quickly. And that's it, right? That's the big difference between glycogen and starch. It's just a good way to store your carbohydrates. So I will come back to this slide in a minute. I'm going to go back to our notes, write some notes here about carbohydrates. Okay, so the basic units of carbohydrates are monosaccharides. And so we might have glucose, fructose, ribose, etc. The linkages we've mentioned are called glycosidic linkages. And what's the function of carbohydrates? Well, same number one as our fats. Food source. We eat lots of carbohydrates. We eat vegetables and fruit. They have lots of starch in them. And also energy storage. We store a little bit of energy in our muscles and a little bit of energy in our liver in the form of glycogen. All right, so I'll come back to this in a moment. Let's go back to PowerPoint. There we go. So it turns out there are tons of things that carbohydrates can do. And the food carbohydrates are just one of the things that can be done. So if you take a look here on the left, I have our starch. And it says here in the note, it says we can link these things in different ways. So there's a different way we can link these glucose residues. And this is unfortunately a very poor picture because it's really not representing what's going on in 3 dimensions. And I'll show you a better image in a minute. This is the kind of thing where molecular models really do show a huge difference. If you take a look at these starch residues, I'm looking at the on the left, the glucose on the left. They're all in the same orientation. So this one and this one are both, what shall we call them, upside right. If I look at the one on the right, this one is upside right, and the second one is upside down. So you might think, well, what does that matter? Well, I'll show you in the next picture why that matters. But what we really are is we're getting, on the left, we're getting starch, and on the right, we're getting an entirely different carbohydrate called cellulose. So this is called, on the left, an alpha linkage, and on the right, a beta linkage. Again, you'll hear more about that when you take biochemistry. So here's a little bit of a better picture of what happens when you link these things, one upside down and one upside right. You get all of these things here. So if you take a look, these little dotted lines, right? These little dotted lines are representing hydrogen bonds. So you probably learned about hydrogen bonds in high school, maybe briefly. I'll tell you they are weak molecular forces. And so what? So they're weak? Well, it turns out that now we have hundreds and thousands of hydrogen bonds. So even weak forces can become strong if you have lots of them. So there's way more hydrogen bonds in cellulose than starch. And so it makes cellulose a much more, it's less water soluble, it's less digestible, it's a much tougher substance than starch. Starch dissolves nice and easily in water. When we eat it, it breaks down in our stomachs pretty easily. Cellulose, we can't even break down cellulose as animals. We just don't have the enzymes to do it. Here's the picture from the textbook. You can see it's not quite as nice as the one I just showed you, but there are, circle a few, some dotted lines here and there representing those hydrogen bonds. Not quite as nice of a picture, I think, but you can see it's forming these fibers, and so you can see down in that yellow image at the bottom right here, it's making all these little fibers, and those little fibers are part of the plant cell wall. So as I mentioned, animals cannot digest cellulose, and it turns out a huge part of the plant world is cellulose. And so how do we digest it? Well, here's a cow. You probably know a little bit about cows. They have extra stomachs. They have like 4 stomachs, and they chew their cud all the time. And lastly, they have a special type of bacterium that helps them to digest the cellulose. Without that, they would not be able to eat grass and hay and all the other stuff that we feed them. They wouldn't get any energy out of it. Okay, so I'm going to go back to our notes here for a second, and I'm going to add functions. We have cell walls. And so I'm going to add some carbohydrates here that we're going to talk about. So we just talked about cellulose. We're going to talk about chitin in a moment, and we're going to talk about something called peptidoglycan. So we'll talk about that, what that is in a moment, and those are part of cell walls. So by the way, feel free to ask me questions at any time using the chat function. I'll probably see it eventually and be able to answer your questions. I know at this point in the semester thing, probably much of this is review. It will eventually get a little bit more complex. Okay, so back to our PowerPoint. So I had mentioned something called chitin. So this is chitin, or at least this is the subunit of chitin. And what I'm looking at here, this just looks a lot like glucose. In fact, it is. That is glucose. It's glucose, and then what we've added is this group here. This is called an N-acetyl group. So we call this N-acetylglucosamine instead of glucose. But it's basically glucose. We put these things together in a very similar manner to glucose, and you're going to get something called chitin instead. So I just had a little question. Somebody says, can you go a little slower when we do the notes? Don't worry, I will go back to the notes. And just let me know if I'm, once again, when I get back there, I will be toggling back and forth between them. So where do we see chitin? We see chitin in all sorts of places. It's part of the fungal cell wall. If you know anything about insects, they have an exoskeleton, and chitin is part of that hard exoskeleton. And chitin is also used to make some sorts of surgical thread. So it's very tough, not digestible substance. So here's that peptidoglycan. We're going to talk about peptidoglycan in a little bit more detail when we get to bacteria. Take a look at that word, right? We've got peptido. So that means peptide or amino acids. And we have glycan. So glycan is like glyco, that means carbohydrate. So what do we have here? We actually have a carbohydrate structure. We have this, these two carbohydrates, one called NAM, that's N-acetylmuramic acid, and also NAG, N-acetylglucosamine. You don't need to know those words, by the way. I'm just showing the structure to you to show you that here again, we have these structures that are basically glucose, but they've got some extra modifications on there. So again, we've got an N-acetyl group over here, we've got an N-acetyl group over here, and this NAM also has these things on it. It's alanine, glutamine, lysine, alanine, and alanine. So these, you probably know, maybe you've heard those words before, those are amino acids, so that's a little peptide. So peptidoglycan, what the bacteria do is they link these carbohydrates together. And so here's the carbohydrates right there, all linked together. And these little peptides make these little cross bridges. So you can see right here, just draw in a few cross bridges. There we go. They link the two chains. So rather than just having these fibers, we actually have a really strong mesh-like structure. And this is what makes the bacterial cell wall. So we're going to come back to peptidoglycan when we talk about bacterial cell walls. And it's an important structure to know a little bit about. So that word there, peptidoglycan, it's a good one to know. You'll need to know it for the midterm eventually, so I thought I'd introduce it now. Okay, so it says our cell has something to eat. So there we go, we put some food in there. There's some carbohydrates or maybe some fat, and we also have carbohydrates used for the cell wall. So I'm going to go back now to the notes, and I had one more thing to add about carbohydrates, which will come up when we talk about membranes. And I just don't have a slide for it here. But carbohydrates, I'll put this note here, glycoproteins and glycolipids. I haven't really talked about what that means, but a glycoprotein is a protein that has a carbohydrate, and a glycolipid is a lipid that has a carbohydrate. And so I'm going to say these are used in membranes, and I'm going to call them as cell markers. Okay, so this is kind of jumping ahead in the lectures a little bit, but I just thought I'd stick it here to make your notes complete. Carbohydrates are found on some of your cell membranes, and they're kind of like little baggage tags. So they're saying that this is a red blood cell, or this is a liver cell, so, you know, this is important with multicellular organisms that we have mechanisms for our body to recognize what its different parts are. So I'm going to call them cell markers. I don't know if there's actually a better term than that, but if I find one, I will include it here. Okay, well, maybe what I'll do just to make these easy to read is I'll bullet point them, and that way you can see them a little bit better. There we go. Okay, I'm going to switch back to the PowerPoint here in about 10 seconds. A little warning this time, and I can see we're almost up for time. I'm just going to introduce maybe proteins, and then we'll come back to that next day. Okay. So I was going to do another Kahoot. I'm going to save that for next day because I do want to introduce proteins. So we'll just come back to that. Okay, so proteins are the next group of macromolecules. I'm going to skip what they do and come back to that next day. What I did want to do for you is just introduce the chemistry of proteins, which is really the basic chemistry is amino acids. So an amino acid looks like this. I'm going to draw one up here in the corner. What you do is you have a carbon, central carbon, and let's see here. I'm going to actually draw it the same way they have it. I was going to draw it a different way, but I'm looking at how they have it in the textbook here. That carbon is connected to a hydrogen. That hydrogen is not doing much, but carbon will have extra groups on it. And then it's going to have a nitrogen connected to it. And sorry, this is just, I'm really slow at drawing with my mouse. Really need that pen again. So this thing here, NH2, so this is called an amino group. There we go. And then over on the other side, we have a COH. Sorry, I should have left a bit more room on this slide to draw this. And this is called a carboxylic acid. So sorry, I don't have quite enough room there, but I'm just going to squeeze this in. Carboxylic. Maybe I'll draw a better one for you next time. If I can get a hold of my pen. Okay, carboxylic acid. There we go. So you can see all those amino acids down there have that basic functional structure. And then lastly, I'm just going to call this R. Why R? Because there's no atomic element with the letter R, and that's the side chain. So all the side chains here are shown in yellow. So our amino acids, they all look like this, right? They have the amino group, they have the acid group, and then they all have these different side chains. If you take a look, this is showing some of them. There are 20 amino acids, glycine, alanine, valine, and so on. Usually they end up with ene on the end. And some of them are hydrophobic. That's what this slide is showing, is all these hydrophobic ones. Some of them are polar, and so that means they are water soluble. Some of them are charged, so they have positives and negative charges. So mostly I wanted to just introduce the proteins. I'm just going to switch back to the notes for people here. I'll fill in these notes here, other than I will just say the basic unit of proteins are amino acids. So that's pretty much all I have for you today. Like I said, I will be sending out the lab instructions tomorrow. Hopefully by noon is the plan, and you'll have some time to work on lab 1. If you look at the lab schedule, there will actually be nothing due for 2 weeks for the labs, because lab 1 and 2 is going to be a combined assignment report kind of thing. And so you'll be able to work on lab 1 though, of course, and get some of that completed. And then when you get lab 2, you'll be able to put together the full report. So I'll also talk a little bit about report writing as well. So if there aren't any questions, I'll end the lecture there. Oh, I see there is one question. It says, do we need to do anything in preparation for the lab? I would encourage you to take a look at that introduction to the lab manual that I sent out last week. I think I sent that out on Friday. If you don't have the email, it is on Moodle, and that's all you need to do for this week. Will this PowerPoint be posted on Moodle? Yes, I meant to do that this morning and forgot, so I'm going to work on that and get that up hopefully in the next 10 minutes here. All right, I'm going to end the lecture here. Everyone, I hope you have a wonderful afternoon.